Hello, my name is Caitlin Mills. I'm a PhD candidate in biostatistics at the Keck School of Medicine at the University of Southern California, and today I'm going to talk about my thesis work. Human beings are complex organisms with many different types of cells, tissues, and numerous stages of development. We used to think that because of this complexity, we must have a lot of genes. But when the human genome was sequenced for the first time in 2001, scientists were very surprised to discover that humans only have about 20,000 protein coding genes, which is only 2,000 more than C. elegans. And in fact, these genes make up only 1 to 2% of our DNA. So what could explain the level of complexity found within the human organism? If you think about the primary function of protein coding genes, it's to encode proteins. Proteins are the gene product. But each different type of cell has a unique gene expression profile, a different set of genes whose products are needed, but in varying quantities and at varying times. Genes that are not needed for that cell type will be off, not producing any gene product. The process of gene regulation is how these intricacies are achieved, and a big component of gene regulation is the enhancer. Think of an enhancer like the dial, determining the level of output of a gene product. We tend to think of genes as being either on or off, but once the gene is on, there is also everything between minimum and maximum production. And when you consider that for each gene, there can be multiple enhancers regulating its production levels, and each enhancer can regulate multiple genes, and that in each cell type, these relationships can be totally different, and when we factor in that there are about a million enhancers regulating these 20,000 genes in a cell type specific manner, we can quickly see how a mere 20,000 genes can be the basis of a very complex system once a million enhancers become involved. And these enhancer gene regulatory relationships are quite important, not only for their development, but also for the healthy maintenance of our cells. If something were to go wrong with an important enhancer gene regulatory relationship, it is easy to imagine how badly that could affect the cell population. And indeed, enhancers have been associated with many serious diseases, including many forms of cancer. Unfortunately, although we may be able to associate specific enhancers with specific diseases, for most enhancers, we do not know their regulatory partners. And if we do not know which genes an enhancer is regulating, or should be, it is very hard to explain exactly how associated enhancers could be contributing to disease pathology. So how do we determine the target genes of these enhancers? Well, we could examine each enhancer at the bench in all cell types of interest, but this would be exorbitantly expensive and time-consuming. Another option would be to utilize computational methods to help us predict which enhancer gene pairs have a regulatory relationship. To do that, we use machine learning. Because there is already a lot that we know about enhancer gene regulatory relationships, we can describe the characteristics of enhancers regulating their target genes and teach the machine how to spot the complex pattern of what is different between these enhancer gene pairs and enhancer gene pairs that we know have no regulatory relationship. If we can teach the machine how to distinguish between real and false enhancer gene regulatory relationships, we can predict a large number of enhancer gene regulatory pairs with high probability, and if any of these enhancers are associated with disease, it can help to better understand how they contribute to the disease. This information could even help to find novel therapeutic targets.